Hello, in this video we will discuss about the base excision repair and the single strand break repair. So in this video we will just understand about the DNA break. So the base excision repair we will discuss and single strand break repair, the single strand. So it's the short patch pathway we will discuss. This short patch pathway basically we will learn and how to repair the DNA. That is the major target to understand in this video. So this is the nucleus contain chromosome and the cytoplasm is here and nucleus and plasma membrane. So this is the cell. This is the somatic cell, remember. This is contain DNA. DNA contain nitrogenous bases adenine with guanine, adenine with thymine and guanine with cytosine. So here is a different situation, the apurinic or apyrimidinic AP site, which that will be a base excision, and this is the base excision. And after the here is the single strand break, and this is the damage base. Damage base due to some reason, due to some epigenetic mutation. So remember, and after this the recognition of AP uh, uh, recognition of AP site, apyrimidinic site, which that is basically our single stranded base strand and DNA damage signaling. This DNA damage signaling will lead to recruit through a FUS. This is the recruitment protein which that recruit the APE1. APE1 is the ape endonuclease 1. For example, the apyrimidinic site endonuclease 1, which that will be recruit through a FUS is the protein, and after this the PARP1, this PARP1 which that is recruit, and after this binding to the basically is the app site, the apyrimidinic site. So in this way, the first also recruit the XRCC1 protein for the repairing protein and the perilytic PARP1, and this is the term basically is the PNKP is the polynucleotide kinase 3 prime to phosphatase enzyme, and the TDD1 and 2 is the tyrosine DNA phosphodiesterase 1 and 2. So in this here you can see this is the basically the here you can understand this is a DNA glycosylase enzyme used for the catalyze the removal of the damaged base through cleavage of the C N glycosylic bond, leaving an ape site. Ape site means the apyrimidinic or apurinic site. So after this, the AP1, ape endonuclease 1 and PNKP, the polynucleotide kinase 3 phosphatase enzyme, and the 2, the aprataxin, which that is used to the hydroxyl group is here 3' side, and the 5' side is the phosphate group. And in this way, here you can see the ORDRP, which ORDRP, which that is recruit. And in this way, you can understand this is the DNA ligase enzyme recruitment, and the, DNA ligase 3 and XRCC1 stimulate ligation through first recruitment. This first protein is recruitment after the signaling of the DNA damage, and the DNA polymerase B, beta, and XRCC1 combine together to form a polymerization, and the DNA ligase enzyme used for the ligation of the gap. So this is a base excision repair. While the double strand break repair, we will let's learn about the whole double stranded break. This was the single stranded break, but let's begin to understand the double stranded break. So the homologous recombination and the non-homologous end joining, which that is the two different terms involved for the double strand break repair. So in this way, here is the mRN and CTIP, which that is recruit to the damaged site. After the damaged site, the RPA, the replication protein A, which that recruit after the recruitment of mRN and CTIP. After this recruitment, basically the RPA, the replication protein A. This replication protein A is recruitment. After the recruitment, the replication protein 51 also combine, and the BRCA1 and BRCA2, which that is also recruit. So the BRCA1 gene, which that is produce this protein in the response of DNA damage. So remember, so here I am drawing the again the RAP51, the replication protein A, replication protein 51, not A, which that is combined with the BRCA1. This BRCA1 is the gene which that is transcription to translate due to the signaling of the damage of the DNA. So the double-stranded break down, the break repair signaling, which that recruit the BRCA1 and BRCA2, and it is also involved for the breast cancer. So in this way, the BRCA1 when not recruit, so the breast cancer can cause. So here is the XRCC3 and the RAP51, the replication protein 54 and 51C, which that is recruit as well as on the other hand. So remember that the basically the another strand, because it is the homologous recombination, so the another strand will recruit same which that is a trans look like, and this will which that is lead to polymerization, and the repair will be complete, and in this way the homolog template will be separate and the restore genetic information that was disrupted by the double-stranded break. So in this manner, the next is we will discuss about the non-homologous end joining. In this way, the homologous template strand will not involve but directly repair, because it don't need to template use. So here you can see the double strand break will lead to recruit the Cu70 and 80 heterodimer formation, and in this way the DNA P protein kinase CS, which that will recruit and bind the site of damage, and kinase activity will lead to Artemis recruitment, which that will and DNA polymerase recruitment will lead to polymerization of the DNA after the stabilization of the break. So in this way, the XRCC4 and XLF and ligase enzyme 4, the ligase enzyme 4, which that will recruit, and the basically combination will lead to rejoining and reaction. Rejoining reaction will lead to repair the DNA. But here is you can see the XLF and XRCC4, which that is involved, and ligase enzyme involved for the ligation after the complete polymerization to nucleotide formation, and the gap will be repaired through a ligation. So in this way, this is the damage. If damage will not repair, so the cancer causing gene activation or other damage and disease causing effect can involve. So remember that things. In this way, the base excision repair and single strand break repair as well as the double strand break repair we had discussed in this video. So next video we will also learn about another type of breakdown of DNA and their repair. So that is the double strand and single strand break repair through short patch pathway. So thanks for watching. Please make sure to subscribe, like and share if you like this video, and comment below for understanding the better. So thanks for watching. We will see in the next video. Bye.